Hello, everybody. Can you hear me?
All good, Anna. Keep going.
Okay, so thank you for coming to this open session to the university students, and welcome to this event. My name is Ana Mena. I'm one of the organizers of this event. Vanessa Borges and Rita Caré are also in the team that is organizing the event, but we are also counting with many IGC scientists and coordinators of programs in this organization of the event. So you will be also having the opportunity to talk to each one of them. So I am not sure if you are all listening to me. Can you please tell me? Yes. Okay. Yes. Because sometimes you freeze, and then I'm not sure if you are listening or not. So I'm going to just share my screen with you guys. You know, usually we would be hosting you at the Gulbenkian Institute of Science, and I'm going to refer to the institute as IGC. So we would be opening our doors and having you there to visit our labs. But today, due to the pandemics, that's not possible. And therefore, I would like to invite you to have a quick look at the IGC of where we are. So we are located in Oeiras, a small town very close to Lisbon, about 20 kilometers to the center of Lisbon. It's a very nice neighborhood close to the Atlantic Ocean. And now you start seeing the institute appearing here. And we also have the advantage of being near a palace from the 18th century, and to be surrounded by these very beautiful gardens that you can see here in the video. And here is the IGC community just getting ready to wave you and to welcome you to the IGC and to this session. So for those of you who speak Portuguese, we also have a virtual tour of the Institute that you can do. And so this tour, it was made using 360 degrees video technology. So you will be able to visit the labs and see all the spaces. And also we have prepared some science enigmas. So you can also interact with us and to solve a mystery that we have included in this virtual tour. So, but what is the IGC? So we are an institute that aims to solve tomorrow's challenges today. And to do that, we are doing discovery-driven research in the life sciences. So we are answering to many different scientific questions. And so we have 33 research groups working in different areas of biology. So we are a multidisciplinary institute, but we also are working in a very cooperative way. So our scientists, they interact with each other, and they are supported by cutting edge technology that is also being implemented by experts in our facilities. Also, we aim to make science as open as possible, and therefore we are developing a range of activities to trying to take science to the heart of society and to promote public engagement and participatory initiatives of citizens in science. For you to have an idea, we are about 390 people working at the Institute, of which 284 are researchers. And we have people from 44 nationalities. As I said, we have core facilities and services that are supporting the 33 research groups that exist at the Institute. And perhaps the part that interests you most is that we have dedicated programs to support postgraduate education. And these programs we have at different levels. So we have programs for predoctoral training, such as summer schools, and students can also do the master's project here at the Institute. And also we have a PhD program if you want to pursue a PhD. So today you are going to hear about these programs in more detail by Jorge, Ricardo, and Ana Aranda in the first part of this session. And in the second part of the session, you can talk to the scientists and pose any questions that you have about these different initiatives. I also want to call your attention, and especially for those of you who are coming from a Portuguese-speaking African country, or that have a Portuguese nationality, but you descend from a PALOP. There are these António Coutinho Science Awards. The call is now open, and you can apply to these awards to get a fellowship to pursue your studies here, not just at the Institute, but in Portugal. So maybe this is a good opportunity for you to have a look, and you can find more information in our website. Also, at the Institute, we have a training program in bioinformatics that started in 1999. So for many years now. These are short duration courses, and they consist on hands-on training techniques, interactive exercises, and cross-questioning. So all this information that I have been telling you, you can find in our website under the training section. Okay, so now I'm going to now stop talking, and I'm now passing the word to my colleagues. So, Jorge, do you want to start?
Yeah. So, can you listen to me? Can you just wave and say hello? Cool.
Yes.
Good.
Yes,
So,
yes.
Welcome you all. It's really impressive to have 200 people that are interested in what we are doing. So it's really a pleasure to be talking to you. So I'm going to try to share my screen because I prepared a few slides. So are you seeing something like a photograph of the IGC?
Yes.
Okay, fantastic. And now you are seeing some stupid slide that says doing a PhD at IGC, right?
Yes.
Yes.
Okay, cool. So that's basically the first slide, just to check that everything is fine. So we have, at the IGC, we have a long tradition of PhD programs. So let me see if I can change my slide. Yes. So if you see, we started organizing PhD programs with a structure, so courses, then following the time in which you choose a lab and this kind of stuff, that goes back to 93. So that's probably way before most of you were born, which is fine. That's good news, because that means that we have a tradition, but also we are not stuck in time. So if you see, we have been organizing different programs, and this thing in orange is currently our official PhD program in integrative biology and biomedicine, okay? Which I would encourage you to apply if you are interested in solving the problems of tomorrow today, okay? And that's the thing. So part of these programs that we have done in the past were actually trying to recognizing, for example, here, that there was a lack of training of PhDs from medical doctors. So we actually created a program for medical doctors. We created a program for computational biology in a period where there was no one training people in computational biology. And so now the integrative biology and biomedicine program actually tries to solve a problem, which is, oh, by the way, this is just to show you that we are about people. So our main job is actually to try to give opportunities to young people. So this is 2007. This is the retreat of our PhD students in 2017. Okay, and what you see in there is happy, young, diverse people. And that's the most fantastic thing about doing a PhD, and let me say it from the beginning, is that you spend 4 to 5 years being pushed to your limits of your intellect and always interacting every day with people that most of the time are smarter than you, okay? Which you cannot actually imagine another situation where you are always surrounded by smart people. So if anything to do a PhD is a good suggestion, and a good thing is do that, because it will be the best time of your life, okay? And that's just why this photo is here. Now, if we think about why we are organizing this PhD program and the way we are doing, because we are actually trying to solve a serious problem today. And that problem is that because typically the undergraduate training and the needs of research, so the needs of preparing us to solve the problems of the future, they are not really adjusted. So there's a big gap there. And nothing like a COVID to illustrate that. So we typically say the biology is the science of the 21st century, or the 21st century will be the century of biology, like the previous one was one of physics. But actually, if you look at the training of people from exact science and engineering, they are completely, usually clueless about biology. Okay, and I heard a rumor that there are people from technical here, and if you are coming from physical engineering or something like that, we welcome you a lot. You have a fantastic training in science, except for knowing a little bit of biology that typically you are clueless. Okay, but we really love you, so do apply. Now, the other problem is the other way around. So you now look at the people that are undergraduates from biological sciences and medicine and biomedicine, and they are absolutely incompetent with math and computing, okay? And at the same time, we know that the world is becoming increasing quantitative, okay? So we all heard about big data and artificial intelligence and this kind of stuff. This thing is invading our research every day, and our undergraduate students, they actually don't know about computing, nothing. They cannot program. They don't, most of the time they come here with very poor statistics. So that's the problem that we are trying to do, okay? And now, our program, I think it's a unique opportunity, not only because you will be dealing with smart people, but because actually we give you the chance to choose your own project and to do your own research. And that's something that we are very proud of, okay? And we do one thing, which is, so our program, to get in, you actually have to apply to a call, that's typically an international call. And then you get in, you don't get a project, you just get guaranteed a fellowship. So we'll pay you to study, but then it will give you 6 months of courses. And this, I can bet you, one of you, if you have the less than 10 people that we select, will be uncomfortable in at least one course where you are completely out of your domain. It can be the statistics, it can be the cell biology, it can be biophysics, it can be the ecology course, it can be the development course. You will be out of your league, okay? But your colleagues will be comfortable. And in the ones that you were comfortable, they are uncomfortable. And so we count on the students to teach each other in this phase here. But you notice one thing. So this arrow here is just to say that after 6 months is when we give you the opportunity to start thinking about your research project and finding a lab. A lab that will be inside IGC with some of our researchers. But that's after going through the courses that you actually choose your research. And then we give you 4 to 5 years to complete your research. And why are we doing this? Okay, because we count on you to choose a project here that's in the limits of our knowledge. So it's actually going to the place where you don't know nothing. That's what we expect you to discover. That's what discovery-based science means. You will go to a place where no one nobody has been before. So there is no comfort. There is no place to attach and to feel, except one thing that is fantastic at IGC is the scientific community that will help you. Okay, but that's why I want to say that. And now, this slide here is just to show you the trend in Europe that the PhDs are not the way that we are doing. So typically it's 3 years where you have to, once you start getting and you get out, you get in with 11 the project, and you have 3 years to complete your PhD. So 3 years is very tough to do something that is really new and really a discovery. You cannot take any risks in 3 years if you are guaranteed to get your thesis in the end. The only way to do this is that you do what your boss tells you and you become a research assistant. We don't want that here, okay? So we are looking for people that actually want to take risks with their life. And what we do in exchange is we give you a cocoon of scientists that are committed to make you do the best science that you can, and a community that will help you. And then we'll give you a fellowship for as long as you need until you finish, okay? And that's one of the great things of the Institute and doing the PhD here. Now, what are we looking for? You can imagine that if you are going to invest this amount of money, people, and time in a student, we want to have brave students, people that are independent thinkers and that they want to get out of the straitjacket of their undergraduate specialization. The people that feel comfortable venturing into the unknown, okay? And that's who we are looking for. So you apply, you write a motivation letter, that's a very important thing. You send reference letters, people that will contact for you and will tell us about yourself. And then what we are going to do is to interview you. And in the interview, we'll try to get into your high potential. And that's what we'll select you. If we think that you have a high potential, okay, that we can bring you into our family, okay? Now, a few stupid slides. So courses, of course, we have courses, and we are a research institute. One of the things that if any of you comes here that you will realize is that your teachers are not university teachers. They are researchers that will be telling you about research that they did last week, okay, most of the time. That's what will happen to you. So that we have something like hundreds of teachers that we invite to come and teach the students. I don't know if you can see a video here. That little guy over there, that's a guy called Scott Gilbert. If any one of you ever got interested in developmental biology, you got one book that's called The Gilbert, okay? So that's one of the teachers of our students, in addition to the faculty of the Institute. But you will get exposed to people that normally you would not see. So our teachers are not people that read in books, like the book of Gilbert, what developmental biology is. These are people that, like Gilbert, wrote the book, okay, and did the research that is behind the book, okay? And I'm not going to bother you with kind of disciplines that you are going to have. Now, this is just to tell a slide that's a bit pretentious, and I warn you on this, so which is, I don't know if you heard about Leiden ranking of universities. So there's one of these things that ranks universities, and if you want to know where you want to do, you go and you try to get the top rank. So this is one of the criteria, which is the percentage of papers that the people publish on the 10% best journals in the area of biomed and health. And this is just to show you the neighbors of IGC, which is the Imperial College, the Wiseman, the Princeton. We are in the same ballpark of these things. So what I'm trying to say with this slide, this is a nice way, like the usual thing that we see in the tourism in Portugal, go abroad by staying inside. This is one way of doing that. Okay, you can have been doing research in an institute that's in that range of quality of science. Okay. And I'm doing it on a little bit propagandistic way, and it's pretentious, but that's what it is. And I just wanted you to know this. Now, then what kind of research do you do? So these are papers from our PhD students. You see people that are trying to understand the shape of the vertebrate embryo, people that are doing conservation of lemurs in Madagascar, people that are trying to understand obesity and the relationship between obesity and the nervous system, people that are trying to identify, not only trying, actually identifying antibiotics and repurposing them to, for example, allow you to survive sepsis, okay? Or people that are doing mathematical models for how cancer develops in cell lines or whatever. I'm not going to, and evolution and so on. So you see the diversity here. So that's why we need people with different backgrounds. That's why we are looking for brave people. Now, and what we give you is programs, people interacting with people. This is seminars. This is a typical seminar, crowded with people listening to. Today, as you can imagine, most of the seminars look like this, like the session that we are doing today. The labs are open space. If you visit the open day video, three-dimensional video, you will see that most of the space is open. And so you can interact with your neighbors. We have facilities for computing, or as well as facilities, for example, here is an incubator to have germ-free mice. You know what is a germ-free mouse? It's a mouse that has grown since forever, and its parents also, without any contact with bacteria or viruses or so on. So they are completely isolated in there. This looks a little bit like a NASA spaceship door, okay? Actually to get the food inside and this kind of stuff. So we use this to study microbiota that under a controlled way, or infections in a controlled way, okay? And we have the same way that we have facilities for mice, we have facility for flies, we have facility for plants, we have all sorts of things. Super amazing imaging facilities, and Ricardo probably will tell you about that, or Erin for the electron microscopes. I'm not going to bother you, that you can find all these in the website. The important thing is that you have both the intellectual environment with visits of more than 100 people that you can interact per year. Once you come here and you can talk to them, and also the infrastructure. And the biggest thing of the IGC is this. It's not all the technology, it's the people, okay? Scientists or the person that takes care of the restaurant, that is side by side by the director. And there is diversity here, mutual respect, and everyone treats a PhD student as a researcher, a full-blown researcher. Okay, it's not escolinha, okay, it's not little school, so we treat you as an equal. And so if you want to be a scientist, that's the best environment to be, okay? And this is very supportive. This is a statement from Anna Stankovic, and you will find a video about doing a PhD at IGC. And Anna, at some point, she says in that video a fantastic thing. She says, I always feel that the whole IGC is doing my PhD. Not only me. This means not that we are helping her too much, is that we are worried with her, and we discuss with her all the time, the science. And that's what she felt. Now she's in Europe doing a postdoc, but she still remembers that nice environment. When you talk to her, she misses the environment where everyone knew your name and everyone knew what you were doing, okay, and understanding your difficulties, okay? And that's quite cool. So, and I'll stop with this, but just with the thing of, if you want to do a PhD, come and do it with us. It's a nice place, okay? And I don't know if we have answer questions now or later.
After the 3 sessions.
Okay.
Okay. So if you don't mind,
yeah, I don't
we'll
mind.
open.
Let me stop sharing them
afterwards.
To see if I can actually manage to do that. Where is my menu for? So let me close this one.
Thank you very much, George.
Did I stop sharing?
So please keep in mind the questions that you want to ask to George, okay? And now I'm going to invite Ana Aranda to talk about the master's projects and how you can apply to do a master's at the sorry, I forgot to introduce Anna. She's the head of the training unit. I forgot to introduce you. Sorry.
That's okay. Welcome you all. Well, after Josh's talk, I'm here not to let you know about the scientific background, but to tell you a bit about how the master's project. So if you're still doing your master's, you're not ready to join the PhD yet, and if you can do a master's project within your master's program, then the IGC doesn't have a program as such, but it does offer the possibility for you to conduct a project with one of our group leaders. So it doesn't have a call. Well, it is open in permanence. So if you check on the website, you can have a look at the available master's projects that we have, and they do change throughout the year. Right now, we've just updated the website if you want to check. And basically, what it entails is that you have to be enrolled in a master's program. You have to talk to your master's coordinator to check whether you can do the thesis elsewhere. And then you can then apply by sending an email to have a look at the labs that we have and the projects that we offer. Even if a project is not listed, if you really wanted to talk to a specific researcher, we encourage you to send an email. I would ask you to send with the CC to these MSc projects. So this is an email address that manages this, and all you have to do is send a motivational letter and a CV, and it's open in permanence. We've just updated some of the projects, and I will share now if I can. So you have here, and actually after this main session, you will have a small break, and I believe that you will be able to stay in this main session for a small presentation from these projects, and you will be able to speak to some of these principal investigators from these projects in case you're interested in applying to one of them. So basically, this is what it is, and I'm going to move on, stop share. So you may want to have a look, but I believe that they will be up again, so that if you want to check again, then I'll stop share, and that's all I have to do. So please contact us if you have any questions, MSc projects, visit the website. And I will pass it on to Ricardo, who will be speaking about the summer school.
Good.
Thank you, Anna.
Sorry,
Ricardo. So Ricardo is one of the coordinators of the summer school. He's also a principal investigator at the IGC, and now he's going to talk a little bit about the summer school.
Can I just confirm? Do you see the summer school slide on screen right now?
Yes, we do.
Yes.
Okay. All right. Sorry. Somehow, if I put this on full screen, it goes to a different screen. So hi, everyone. I'm a group leader here at the IGC, and I just recently joined, although I started my career here more or less on the stage, or at the time that you're in. What I mean with that is I came to the IGC to do a small internship when I was finishing my graduation. Interestingly enough, I trained as a physics engineer. And I became increasingly interested in biology, and this was an opportunity to explore a field that was still foreign to me. And as George was saying, the IGC is such a welcoming place that it suddenly started becoming what I wanted to do and the career I wanted to go for. And today I am very much a physicist that uses the background that I have to address biological questions, and often that brings us to a different angle than what we're used to. So how do you get to have a glimpse about this? Well, the summer school is actually a great way to do it. We run it every year.
Ricardo,
yes.
Sorry, we are seeing your desktop, not the slides.
Not the slides. Hold on.
No, we can see the slides. It's just not full screen like you were explaining, right?
Yeah, hold on. Let me try to put it. Bear with me.
Select the other screen.
Hold on. And
Ricardo, if you just swap displays, it'll work.
Yeah, I'm doing it.
There you go. Perfect.
Is that working? Okay,
Yeah.
Sorry. The problems of having two monitors. Sorry. I was telling you, the summer school, it's run every year, and it's actually a great way to get to know the research that is happening at the IGC. This year, we've tried to plan it in a way that you could actually hear from students, group leaders, and even researchers that have experienced time at the IGC, but are not necessarily at the IGC anymore. So you'll get a full glimpse of not only the research that happens here, but what being at the IGC might mean for your future career, and how does that experience blossoms into the next steps that you take. So pretty much anyone from any field can apply, and we welcome diversity. This is something that, as you've realized, is quite important for the IGC. The summer school is going to be divided by two different parts. One is just online classes, and then there is the opportunity to actually have a physical presence internship within the institute for a project that runs between 2 weeks and 6 weeks. The classes occur in the last week of July, and then the small internship happens in August. For the classes, there's going to be a selection of 16 students to join it online. But after they have occurred, all the seminars and all those classes will become available to anyone that wants to see them a posteriori. It's going to be an excellent opportunity to engage with local researchers and to get hands-on experience on even doing a little bit of research. Of course, there is a risk here because we don't know what's going to be the situation with the pandemic in summer. So we're taking it lightly, and we're going to do everything we can to ensure that everyone is safe if you come in for small projects. If we feel it's not safe, then it will have only online classes, and we might not have projects available. But it's something that you can apply, and as things progress, we'll try to decide if it's safe to go ahead and have that physical presence or not. So have a look. The website is now open. You can already make the applications. And it's really a great way to spend a week with us and hear about the research that we do, hear about what's really the impact of doing research and how that has played out in the careers of many scientists and even general people that have used that experience as researchers or in IGC in their careers as a whole. And that's it from my side.
Thank you, Ricardo. So now we are going to open the floor to questions. So if you have any questions that you would like to make to Jorge, Ana, or Ricardo about the PhD programs, the masters, or the summer school, now you can have some time to do that. Okay, so Duarte Candeias, do you want to turn on your camera or not?
My camera is already on.
It's on.
So
okay. Sorry, I wasn't seeing. Sorry.
No problem. I wanted to ask to Jorge, do you have a preference for PhD applications from students who went abroad on their masters or did Erasmus program, or do you just accept everything?
We accept anyone. The only thing that we are looking for, it's precisely that you are competent, knowledgeable, curious, and an independent thinker. Okay, that you can actually engage in a dialogue when you are interviewed, and that's essentially what the most important things that we are going to appreciate. We don't care if you are a physicist or you have a degree in molecular biology. Okay. Of course, we'll try to do one thing that you can imagine. which is to try to get a team that can work together using diversity. Okay, so there is a little bit of matchmaking, as long as that doesn't harm the choice of the person. Okay?
Okay, thank you very much.
Anna?
I'll take an opportunity, since I'm talking about the PhD program. I don't think I mentioned that the call is going to, in principle, open next week, which is not a very important date. The most important date, and that's fixed, is that it will close on the 9th of April.
Thank you, George. So Michaela also has a question. Go ahead, Michaela.
First of all, good afternoon. My question is going to be directed to Ricardo. I am a student that is finishing my bachelor degree in biology, and I have potential interest in wanting to try the summer school. I would like to know what type of candidates you expect to see in terms of CV, if you expect someone that has already done workshops or had lab work in the area, or if you accept even people that are still trying to build their curriculum.
We accept anyone, Michaela. The thing that we'll be paying most attention is to your motivation letter. We want to hear from you how you feel the summer school will be important to you and what you expect out of it. And that is going to be the key factor that will lead us to then choose the 60 students that will be directly with us during those initial classes, which students that won't be selected, we will later on have access to them. So it's mostly about us knowing that that the summer school will be beneficial to you, and if we feel that is really of importance, we'll make sure that you're selected.
And could you also tell me how many students you are accepting, please?
So 16 for the online classes, and then we have 5 projects. Which means that there will be 5 students for those 5 projects.
Okay, thank you.
Any more questions?
I do have a question, but I did forget who did the presentation on the master program.
Anna Aranda. Anna,
hello, Carolina. How are you?
I'm actually.
You're
I'm
okay?
good. Actually.
Sorry, just on a note, Carolina was here for the last summer school, so welcome back.
So I talked enough so she would remember me this year. I wanted to ask if if I get to the second year of my master's degree and I want to do the thesis in IGC, but I don't want to do that specific thesis theme that IGC proposes, is there any possibility that I talk to any of the principal investigators leading the program so that we can try and, try another thing?
That's exactly what I was saying. I would recommend you to have a look at the research groups that we have at IGC and to see what they do and what your questions will be for your project, and to see what would be the most appropriate group. If they have not listed a project, I would send them a motivation letter with your CV and explaining what you're wanting to do and why you want to do with them. Okay, that would be your motivation letter. And based on that, they can invite you for an interview if they have an interest or if they have a space in the lab. So the fact that there is not a project, we are open for spontaneous applications. However, sometimes also the research groups already are looking for somebody for a specific project. So the fact that it is not listed there, it doesn't mean that you cannot approach them. If you have a specific question that could be answered by a specific group at IGC. But for that, you have to have a look at the research groups.
Okay, thank you.
Carolina, can I give you an advice?
Yes.
Okay, this is not, I'm not going to say that the researchers of the IGC are pricks and arrogant people, but they do receive a lot of anonymous emails. People that write a letter saying, I'd like to work with you. But then often those letters don't have anything to do with research that the person is doing. Okay, so you have to be very specific. And I would recommend you to put Anna on CC, because if the PI, the person that you wrote to, happens to just overlook your email, she can then call the attention to that email. Okay, because what you have to realize, particularly in times of COVID, we do often overlook emails, unfortunately. Okay, and it's just natural. So just try to seduce people and put Anna on CC such that she can put a word. Okay.
Okay, thank you.
You're welcome.
Okay, so Alexandre also has a question. Alexandre Belo.
Yeah, hi. It's a question to George. So I'm currently studying in the UK, and the UK has a different system than Portugal has normally. So some people can jump straight ahead from an undergrad project to a PhD project. Mm-hmm. And I was wondering if this is a similar case here with the IGC.
It's complex. We'll have to analyze the case by case, because typically, because we are we don't confer PhD degrees ourselves. So we work with the university. Okay. And the universities in Portugal now, because of the Bologna Agreement, require a master to do the PhD. Now, if you happen to be a person with an exceptional track record until now, we might propose that to the scientific council of the university, such that they can consider your case. Okay. So, for example, if your degree is 4 years, that will be very easy. If it is one of those bachelors with just 3 years or less, then it's going to be very hard to convince the university to accept it.
Yeah, that's also tricky because, for instance, Scotland and England also have different projects. In Scotland is 4 years, in England is 3. I'm studying in Scotland right now, which is 4 years, but it's equivalent to the 3 years. So
but we actually, what matters is the equivalence that will be granted by the university. And so far, they have been treating a 4-year degree as if it would be a pre-Bologna licenciatura that would allow you to apply. So my recommendation is do apply, because in addition, you will get the experience of writing a motivation letter and so on. We'll very soon let you know if we cannot recruit you. Immediately it's saying, I'm sorry, but you will not be eligible this year, try next year or something like that. But you will have the experience. If we get impressed by your motivation letter, we'll even bring you to the interview so that we get to know you better.
Perfect.
And we'll go the extra mile if we decide to recruit you to convince the university.
Okay, sounds good. Thank you so much.
You're welcome.
Okay, so we have one question in the chat for Ricardo Henriques. It's from Inês Gonçalves. The question is, the summer school is just for bachelor students, or a master student can also apply?
A master student can also apply. It's meant to be for pre-PhD students, but if anyone within that range is welcome to apply to it.
Okay, thank you, Ricardo. So Melanie, she also has a question. Melanie, go ahead.
Hi. So I would like to know that when you open the PhD next week, I was wondering if you'll list the list of projects that will be available for the students.
No.
No.
In case you haven't, you arrived late or something. That's something that we are very proud of. Is we recruit students without a project, without a lab. So only after 6 months, we give you the opportunity to start thinking about your project.
But will all the labs from the IGC be on the PhD?
So my recommendation would be, we typically only define that later.
Okay.
So we cannot, we are not prepared actually 1 year in advance because that's essentially what we are talking about. It'd be 1 year in now saying who will be available. In principle, any PI is available except when they have a clogged lab or if they are leaving. So the clogged lab is very easy. If you see a group that has something like 3 PhD students, 4 PhD students, it's very unlikely that will be accepting a PhD student the next year. You see what I mean? Because it would be just crowded the PhD students. We cherish a lot that our PhD students work directly with the PI, not with a postdoc, not with another person, because that's the PI that is our star. Okay, so. And we want the PI to have enough time to dedicate to the person. Okay. So again, the same thing that I was telling to Alexandre, just submit your recommendation letter. Explain if you are really, really interested in one person, say, I would be really, really interested in this person. Okay. Yeah. But don't forget one thing. If you do that, try not to be too specific.
Okay. Okay, thank you very much.
So, Catarina Coelho, you also have a question?
Yeah, hello, I am Catarina, and I'm doing my master's degree in veterinary medicine. And I would like to know if you are going to have scholarships for investigation, because sometimes you are recruiting people to do that, and I'm seeing here in your website that you don't have any positions open, and I would like to know if you are going to open some positions for us to candidate.
But what do you mean, as a PhD student or just to do a?
I am thinking if I want to do a PhD or if I just want to do internship first to see how it goes, because a PhD it's 4 years, and I would like to do internship before to see how it is, because I'm doing an internship in a laboratory with a pathologist for it's my thesis, and I would like to do one in the Gulbenkian Institute to see how it goes.
So as you were saying, so we often the labs open positions for anyone that can apply. But I'll take also the opportunity to say anyone that we admit to the PhD program is guaranteed a fellowship. Okay.
Okay.
So that's for everybody. So once you get admitted, we'll get you a fellowship until the end of your PhD.
But do you think that it's better for me to candidate to the PhD or do first internship with you? Well,
it depends. If you, for example, the kind of internship that you are doing now is not really an experience in research, you don't lose anything in taking an internship in doing research just to see if you like it, because research is not for everybody. It involves some major dedication, and so if that's the doubt that you have, why not go doing an internship? If you know that you want to do research, my recommendation would be don't waste time.
Okay, because I'm actually doing a research with immunohistochemistry in tumors, mammary tumors in cats, and I think that it's amazing what I'm doing because I'm discovering some things that I never saw, but I really would like to know what it's like to be every day in a laboratory, be with people that can teach me something that I don't know, and I don't know. I think the PhD program that you have, it's very good, but in the recruitments that I see, they seem very good too. So that's my question, because I know that to enter in your PhD, it's very difficult. And
well, it's a bit competitive. So we have typically less than 10 positions for more than 100 candidates. So it's tough, but you don't lose anything. As a friend of mine used to say, the no, you already have it. If you understand what I mean.
Okay.
If you don't apply, you will never know. So that part of the competition is just, I think you will have to sort yourself if you feel that you are sufficiently motivated to do science. And my argument would be, if you know that you want to do science, don't waste time with an internship. You will be just wasting time.
Okay. Thank you so much for helping me.
You're welcome.
Okay, so we have some more questions in the chat. So Katia Luz is asking if there is any IGC scholarship for students that wants to start a master program in Portugal. Katia, so if I don't know, so if you are not from a PALOP country, at the moment we don't have anything. I don't know if Ananda wants to add anything.
Unless sometimes a group can have a scholarship for a master's thesis, but that's depending on the group that is advertising. So some of those projects, that will depend on the research group, but some may have a fellowship attached, and some, but those will be published on the jobs on the website. So I would have to check that, but there is no general master's program scholarships from the IGC apart from the PALOP, and it's not to start a master's. It's for the thesis again that we have. We used to have in the past the Nova Life fellowships, but at the moment I don't think we have. So I think that's the only possibility, is to look at the jobs and the calls open, and to see if there is a project linked to a master's, for a specific master's.
Yeah, so as Anna just said, we used to have the NASA Live fellowships. At the moment, these fellowships we are stopping. So okay, so we don't have these fellowships open at the moment, but it's possible that in the future we may have them again, but we cannot. We don't know at the moment if it's possible to have them this year. So it's better if we don't count on these fellowships. Okay. So Mariana also has a question. She's saying that she's a first-year undergraduate student and would like to know what are her chances of being chosen for the summer school. Ricardo.
Sorry, can you repeat the question?
So Mariana says that she's a first-year undergraduate student and would like to know what are her chances of being chosen for the summer school, for being the first-year undergraduate.
It's impossible to say what are the chances. What I mean with that is the selection for the summer school is very much based on motivations and how helpful the summer school will be for your progression, potentially as a future scientist. That's the main thing that we take into account. So I wouldn't be fearful of applying, and the application process is actually pretty straightforward. So give it a try, and even for any reasons you are not selected, we'll keep you informed about all the classes that will be made available a posteriori.
Okay, I think I skipped a question, but it was not. Sorry, Adriana. You also had a question here asking if there are any aquaculture groups at IGC.
No, aquaculture specifically, there is no groups. There is there are several groups that actually work with zebrafish and with killifish and other kinds of fish. And so the Institute has facilities for keeping fish for experiments, but specifically on aquaculture, meaning improving the conditions for fish, for example, no. Or trying to import into aquaculture wild species of fish. We don't do that here. I don't know if I answered the question. Okay.
I think you did. So Miguel Mesquita, you also want to pose a question? Please go ahead.
Yes, thank you very much. Thank you for hosting this seminar. So my question is directed to Jorge Carneiro about the PhD program. I would like to know if you encourage your students to go abroad or to other institutes in Portugal to learn other techniques that can complement the PhD training and help their research.
Okay, so as you might have imagined, so the main part of the PhD training is actually the research, right? And so the training is research driven, and if your research requires that you spend a year in Japan doing I don't know what kind of strange microscopy correlate
there,
you will do that. And it happened I'm giving that example because just yesterday there was a paper from one of our students that was published using those kind of microscopy, and she went to Japan to do it. But there is no limit. So there is no constraint in that sense. So it's not something like, but now let me say something important. It's not like an European ITN that you are forced to spend time in another institution. And sometimes those things are artificial and you end up losing time. So whatever your research needs will provide you.
Okay, thank you very much.
Okay, and Rafaela Silva also has a question for Ricardo Henriques. So she is a master's student in bioinformatics and wants to know if summer school can help her in that area.
It can. I mean, there's going to be a broad range of speakers covering even fields of computational biology and bioinformatics. So I'm sure it's going to be useful for you from that perspective, but also it will get you acquainted with fields that you probably never thought about, but that they might end up being incredibly interesting for yourself also. So you will be partially feeling at home, partly a little bit looking at new things that you've never heard of before. But I think it's going to be exciting from that perspective. I'm going to take this chance also because I saw a question from Maria Inês Ribeiro asking about the fact that she's from a background in biomedical engineering, if she would be a good fit for the type of research that happens at the IGC. Yes, actually, you have a few researchers and a few groups that are aligned with some of the interest topics that exist in biomedical engineering. To give you an example of that, we developed optical physics and artificial intelligence technologies to address biomedical questions in viral infection. So there's the institute has such a broad stream of topics that it covers that you will most likely find groups that are able not only to explore the background that you might already have, but also push you much beyond that also with topics that might interest you, but you never got in touch with.
So I think we are going now for the final questions. We have Isra asking Jorge, so he's interested in doing a PhD in oncology and nanotechnology, and he wants to know if it is available in IGC.
Oncology and nanotechnology?
Yes.
So Well, again, depends on what is your question. So we have, I think we are, for a long time we have hesitated on that, but I think now we have a nano thingy facility to create these small ships where you can actually cultivate cells in a special microenvironment. Okay, so that exists. But saying that we are a place where you would be finding your fascinating questions on nanotechnology here, I don't think it would be the easiest place to do. Okay?
Sorry, I also want to know if it is present the research of cancer treatment, or cancer biology, or cancer in general.
Yes, so
like oncology.
If you look at, for example, genome stability, and if you look at cell cycle regulation, centromere control numbers that and centrosome control numbers that can actually affect one of the causes of cancer, yes, there is interest.
Which group of the research groups?
Well, look for, if you want, just go and look at the group of Monica Bettencourt Dias, our director. Look at the group of Raquel Oliveira. Look at the group of Marco Fumazzoni. Look at those labs, and then there is always possibilities that you discover other passions. We are actually very proud of changing the original passions of people to another passion.
Okay, thank you.
We take pride on that.
Thank
to
you
take
so
you
much.
out of your original scene of your taste, such that you can actually taste other things and start liking it.
Okay, thank you so much. Thank you.
Okay, Maria Inês Ribeiro also posed a question on chat, and it's for Jorge or maybe Ricardo. So she's finishing a master's in biomedical engineering and is interested in PhD topics that could use for her her physics background in medical research. For example, her thesis is about radiation dosimetry. What kind of topics or research groups do we have at the Institute that could make use of this kind of knowledge? Could you give examples of projects like this? So, biomedical engineering and physics background in medical research.
I could actually hand wave of what kind of projects a person with that background could do here, talking about projects that are ongoing. But that's easier if you go and see what papers people are publishing. What I could tell you is, without any problem, the kind of background that you are talking, it's welcome. But again, it all depends on the question that you want to ask, what you want to discover. Okay, so it's every time that I listen to something like, I have a training in this, will be that useful? Sure, it's always useful. But what you'll have to find is your own question. That's what we are looking for. And of course, it has to be, it would be ludicrous to come here and to do something that is related to, I don't know what, technologies that we don't have, or research interests that our researchers don't have. So that would be crazy. It has to be minimally match-made in the halfway of the interests of the PIs here. Otherwise, it makes no sense. So yes, I think there is a lot of stuff that you can do in cancer and therapies and this kind of stuff. But don't forget one thing, the research is a basic institute research. So we do basic science. It's not our main motivation to try to find therapies for things and this kind of stuff. This being said, spin-offs exist. So there is people that have changed, for example, the way you diagnose autoimmune diseases, and after the PhD were headhunted by companies to try to work on therapies of autoimmune disease. So it all depends on you. And I really want to insist on that. You have to find minimally, a lot interesting research that is being done here. And that's very easy to browse through the website and see what people are publishing, read their papers. And then if this thing interests you, just apply. then you will find whatever you can do. But drive yourself by your curiosity, not by the tools that you have. You remember the people that says that anyone with a hammer, everything looks like a nail. We don't want that. So we want you to think what you want to use, and then we'll help you come up with the instrument. Our friend Ricardo over there, he has created his own microscopes just to answer questions. But he's driven by the question.
Erin, do you want to add something?
Erin is stuck.
Oh. Erin, can you hear us? Well, then I will make another question, and then I will pass it to Erin again. So we are not going to receive more questions. I'm just going to say the two that are at the moment in chat, and we need to finish this session. That's why we are not receiving questions. But don't forget that we are going to another session now, and you can continue to put your questions to the scientists in that session. Okay. So Pedro Fernandes, he has a question for Jorge, and he says that he's in his 4th year of pharmaceutical sciences, and is thinking that on a PhD, and he loves genetics, genomics, virology, immunology, oncology. So are there people in the IGC team with these passions, and what PhDs are those people doing exactly?
So there is a bunch of researchers at IGC that you could say that are doing virology, that are doing oncology, that are doing all sorts of things. So some PhD students are working on genomics in the context of cell biology. Some are working on genomics in the context, for example, of genome instability. They are preparing now their projects. So yeah, again, it will be your question. It will be what you want to do. So you seem to have broad interests. So we like people with broad interests. At some point, so any of the things that you mentioned, there is independent researchers doing those kind of things at IGC. So then it will be, I can imagine that matchmaking for someone with those kind of interests would be easy. I can tell you the last person that joined us with this kind of profile and interests ended up working on a completely different thing related to the evolution of genome stability itself. So, I don't know, it's up to you, as usual.
Okay, so I have one final question that was sent to me, and it's from Beatriz. She just graduated in biological engineering and did a master's dissertation working with mice and stem cells, so hematopoietic stem cells. And she would like to know if there is any PhD program in this area at IGC.
So you mean a PhD program in stem cell?
So, perhaps Beatriz, is that it in stem cells? Perhaps that's it.
Well, there is no PhD program
stem cells
on stem cells at IGC. This being said, it depends why you are interested in stem cells, as usual. So there's some people that are working quite a lot with hematopoietic stem cells to try to understand thymic development and development of leukemia when there is a problem in stem cells. For example, there is people that are doing developmental biology Well, that's what I do all the time, is to see what a stem cell is doing. It depends on your question. If what you are considering is, I would like to use stem cells, for example, to cure Alzheimer or something like that, I don't think the IGC would be the most suitable place to come and do it. There's plenty of people that are doing research on that. If you would like to understand in general stem cell regulation as part of the biology, yes, IGC could be a nice place to do a PhD.
My name is Erin Tranfield, and I am the head of the Electron Microscopy facility. What you see here is the main entrance of the IGC, and this is not actually where we do microscopy, but these are the instruments we use, and beside it you see some of the images we take from these microscopes. So since we can't do a physical tour, I thought I would just give you a brief tour of where we are. This is the IGC. If you had come to the main entrance, you would have come in through here, gone to the canteen building, and we'd be currently sitting in building 2 for the seminar. Where the EM facility located is under the antibody, the bottom of the antibody building, under number 3 right here. So we're located in the basement floor, centrally within the IGC. And my team is composed of 4 different people. I'm working with 3 lovely Portuguese ladies, and then me, the random Canadian Swiss lady. So as Anna was telling you all, we are a multinational institution, and I am one of those multinational characters. So at the IGC, in the EM facility, what we do is we do a technique called electron microscopy, which really goes down to the lower levels of detection for resolution. There are 2 main kinds of electron microscopy: scanning electron microscopy and transmission. And the best way to show you the difference is to just show you a picture. What we do at the IGC is predominantly TEM. So in transmission electron microscopy, you have images that look like this, and we do a whole bunch of different kinds of techniques. We have 2 different master's projects or potential projects ongoing at the moment. So the first one is around a technique typically called negative staining. This technique, what we do is we use heavy metals such as uranium or uranyl acetate to create a shadow around the cells. So what you see are 2 bacteria here. In this, you see 2 microtubules with the proteins attached. So these long filaments here are the microtubules. And we have a project working on this. So what is this technique and how do we do it? Well, it's really simple. We have our sample in an Eppendorf. We put the sample on a grid in a process that you see here through these series of drops, and we have essentially go straight to the microscope. It's really that fast. You can do the preparation of these samples in about 5-10 minutes to get gorgeous images like this at the end. But what is our problem? Why are we working on this? Well, the problem, if you go to Wikipedia, is that the main staining salt we use, uranyl acetate, is actually a salt made of uranium. And this part I put in bold is the key part here. This compound is a nuclear fuel derivative, and its use and possession are sanctioned by international law. What this means is that anytime you go to order uranyl acetate, one of the most common chemicals we use in electron microscopy, you always get these little warnings, exclusively for customers in Germany and Austria, etc. It's one of the hardest reagents for us to get in the lab and to work with. So the question that is the focus of this master's project is, can we use replacement stains for uranyl acetate that give us the same kind of contrast and imaging as this? And the whole point of this project is going to be to take different kinds of samples like bacteria, like bacteria, or like, cellular structures like the microtubules here, and to stain them to see if we can get similar kinds of imaging resolution and contrasting between the different conditions. So that's just one example. Another example, because we have lots. So this is sort of the overarching workflow of the general processing for electron microscopy. Some of it uses chemical fixation. Some of it uses something called cryo mobilization, which is a fancy term for freezing. The most common technique is this one here. shown just with the dark shadows. And let me walk you very quickly through this because I know I only have moments. So what we do first is we fix with aldehydes, typically glutaraldehyde and formaldehyde. Then we go through a buffer wash into something called osmium. Osmium is a very heavy, dark metal, also very toxic. And we wash again, we use our dear friend uranyl acetate again, that radioactive compound. Then we do dehydration into a resin, and ultimately we embed in a block. So we get the samples looking like this, that we cut in a process like this, that we then stain again on grids, and then that we go to the microscope. So if we come back to this basic schematic I went through, essentially at every single step of this process, there are questions like, what temperature do you do this for? How long do you do this for? Do you do this in the presence of light? Do you do this in the presence of agitation? So like on a rocker or with a motion? Do we add additional components between these steps? Because this is actually the simplest and the easiest protocol. And probably the most contentious is, which buffer do you use? And so this is not one master's. This is probably about 12 different masters or an entire PhD. Each one of these steps here can be optimized and studied in detail. And just when you think you will have figured it out, I'm going to be mean and switch your model. So you'll have to do all of this for plant cells, and then for animal tissues, and then for tissue culture cells. You can see that this can be endless. So to test, or just to whet your appetite a little bit, to show you an example, we have started this in the lab. Of course, there's 1,000 things that we want to do, and we don't always get to do them to completion. So we started to look at 3 different buffers. I can tell you these are the same cells processed at exactly the same time with all the same chemistry by the same person. The only difference here is the buffer. And I think you can see the difference between the nuclear regions in these 3 cells. And if you compare the resolution of your mitochondria between these 3 cells, you can see there's a difference. So this is something that we would like to investigate in more detail to really understand this, so that when our users come to the facility, we can say with confidence, this is the best protocol for your experiment. If you have general questions about what we do at the electron microscopy facility, you can always check out our website. I call this the opportunity for shy questions. And if you ever want to ask in detail, this is the team and these are our emails, and we're always open to discussion with you. And I think at the end, we'll have questions, right, Anna?
Yeah, so at the end, you can go to your room, and people wanting to ask specific questions about your projects, they will get into that room, okay? Thank you, Erin.
Thank you.
So Monica, do you want to start now?
Welcome, everyone. So it's really nice to see you all interested in the IGC. I'm the head of the cell cycle regulation group, but I'm also the head of the IGC, and it's a pleasure to see so many people interested in coming to the IGC. But today I'm going to talk just with the hat of the head of the cell cycle regulation group. And so these are the people of our group, and in our group, we are basically interested in understanding how cells are made, organized, maintained, and regeneration to form many different functions. And in particular, inside the cell, we are very interested in this small structure called the centrosome, which is actually very important to determine many different functions in our cells. And the centrosome, which is the structure here, is important to nucleate microtubules, which are important for cell division, for cell polarity, for cell migration. But also inside the centrosome that you see here, you have the centrioles that also have a different function, which is to form cilia and flagella. And these are very important to actually signal for many signaling events, but also for cellular motility. And so they actually, at this time, in your body, they are playing many different functions. In your eyes, they are sensing the light. In your hypothalamus, they are sensing whether you've eaten enough or not. In your trachea, they are expelling particles, and in your lungs. And in your kidneys, they are sensing fluid flow, and they're involved in many different cell divisions. And because of that, they're also involved in many different diseases. So we know that actually if you have deregulated centrosomes, which can be caused genetically or by virus, for example, you can have smaller brains as compared to the wild type. That's called microcephaly. But also if you have deregulated cilia, you can have many different phenotypes, from obesity to multiple fingers to cystic kidneys, as you see here, and infertility. But also if you have deregulated centrosomes, that can actually generate cancer, at least in mice, and we know that deregulated centrosomes exist in many different cancers in humans. So these structures have been studied for a long time by these very famous biologists for more than a century. But because these structures were very small, they were asking many important questions, such as like, how are they formed? How are they maintained in the cells? What is their function? But they didn't have the tools to address those questions. And it was only in the 15 or 20 last years that we started to have the tools to ask those important questions. And through proteomics, different screens, and bioinformatics, we now actually know that these structures are there in many different eukaryotes. So it's a pathway that actually, it's a 2 billion years old pathway that is there from the last common ancestor of eukaryotes. And so in our lab, we are very interested in principles that could be applied to other structures in the cell, for example, mitochondria or other ones. So how you regulate the number, how you regulate the length and the structures of this structure, how you form them at the right place, at the right time, and how you keep them through time. So these are principles that we should be learning something for these structures, but probably some of these principles would be can be applied to other organelles in the cells. But we're also very interested in how you break these rules. So, for example, how you form different structures like these ones in different cell types, because for different cellular functions, you form different structures. In different organisms, how do they look like, and have they contributed also to evolution? And how are they deregulated in disease, in particular in cancer? And so the projects that I'll be presenting are related to these major questions in the lab. So there's one that I'm not presenting. It's just today one of our researchers actually proposed it, which is related actually to these different structures in the cell. And she asked, she's actually interested in looking at eggs. We've been looking into eggs and how these structures are regulated in eggs. And we've been looking at fly eggs. And she now recently started to work with a fertility clinic. And she's starting to work also with human oocytes and whether the regulation of these structures could be involved in female infertility. So that's a possible project. Just to show you how the people are organized in the cells, we have a group of people working on cancer, a group of people working on how different structures are organized in different tissues, group of people working on how centrioles are made in space and time, and a group of people actually maintaining the lab and helping many different people in the lab. So Anna is working on the human oocyte projects, and she's not here today. Pilar will show you a project, and the other project is Mariana, who is here. And Mariana is in a medical consultation now, so I'm going to very quickly show you the project. So basically in the lab, Mariana has actually done a genetic screen to identify players that are involved in defining exactly how you have this right length. And so the idea is to look at some of these candidates that define the length and how they have always the same length, and choose one or two candidates to analyze them in more detail and understand their function. So this will involve looking at depletion phenotypes and how, what happens to the cells when you remove these proteins, and to do a lot of microscopy to understand exactly what they are doing. We are using a lot of super resolution and expansion microscopy because these structures are very tiny. And so we can do EM, and we do a lot of EM with Erin that you've seen her images, but we also use these techniques that allow a higher throughput, that allow us to see these very tiny structures in bigger detail using these super resolution techniques that we have in the Institute. And then we might be looking also at, what do they interact with, including some biochemistry. So it will be a lot of siRNAs and depletion, microscopy, and interaction. So the other project, Pilar, are you there?
Yes, I am.
Okay, so I'll move the slides as you go. So this is Pilar, who has this other project, and she'll be presenting it to you. And Pilar is a postdoc in the lab.
Yeah. Thank you, Monica. Yeah. So this project, you've already heard about some of the work that our colleagues are doing in the lab that are important, for instance, to control very tightly the length or the structure of the centrosome. And this is because, as Monica has explained to you, if there are some centrosome aberrations, this can lead to some diseases, such as cancer, for instance. What we are now interested on, can you move on, Monica? What we are interested in now is a new project on the lab that is understanding the possible links between centrosome aberrations and aging. And this comes from very recent but kind of isolated studies that link centrosome aberrations and aging. Of course, as you can imagine, we are interested on aging because our societies are getting older, and this is very good, but also comes with a counterpart. That is that aging is the highest or a major risk factor of many diseases that are deadly and debilitating, like neurodegenerative disease, cancer, and now we know, of course, COVID-19. So as you can imagine, the societies are asking us, the scientists, to put a little bit of more of effort on trying to tackle the aging diseases and age-related diseases. And so what some of the things that we already know is that primary hallmarks of aging are actually impacting on the cell function. Monica, can you? And by doing that, what they do is to generate some kind of secondary hallmarks that are kind of counteracting the primary hallmarks of aging, such as senescence. And cellular senescence or other secondary hallmarks are impacting on the tissue homeostasis and are also impacting on aging. This way, what happens is that there is a kind of positive feedback loop that accelerates the process of aging. And it seems that centrosome aberrations can be impacting also on aging, but this is already not completely known. So one of the other questions is how centrosome aberrations are caused. What is triggering centrosome aberrations? Is it possible that also aging is causing centrosome aberrations and that way it's also accelerating the aging process? So we want to understand first how centrosome aberrations in this project are caused, whether aging has an impact on the cause of triggering centrosome aberrations, and also, of course, how centrosome aberrations can be impacting on aging. And we are going to try to tackle these two questions specifically in Drosophila. We use, in the lab, we have people that work on Drosophila, some people work with mice, some others with cell types, with cellular cultures. And in particular, we are going to try to understand this in Drosophila, in this case, specifically on stem cell types. And yeah, if you have more doubts, come to our breakout room and we can answer all of them.
Thank you, Pilar.
Thank you, Pilar. Thank you, Monica. Okay, so now we have Karen and Elisa.
Hello.
If you want to start your presentation.
Yes, so I will start and then Elisa will take over because she's actually the one who will guide this project, whichever project people choose. So this is the cell biology of tissue morphogenesis lab. This is a very old picture because we couldn't update for obvious reasons. What we work on is the development of the vertebrate eye. What we use is the zebrafish as a model tissue. And we have a very general presentation now about the general questions that Elisa is asking. And she has many, many, many more questions than she can ask within one PhD. So there's a lot of things to explore. But if you want to have more detailed explanations, please come to the breakout room because we have two relatively defined projects that we can discuss in the breakout room in case you are interested in the general question that Elisa will present now. Thanks, Karen. Can you hear me? Does it work? Oh, okay.
Yes, it is.
Sorry, wait. I'll move this on the side. Okay. So as Karen mentioned, in the lab, we're interested in understanding how one specific part of the eye, the retina, develops. So the retina is this beautifully layered structure, and it is part of the central nervous system. And as it is part of the central nervous system, it is composed of neurons. In particular, there are 6 different types of neurons that occupy a distinct layer in the retina. But what is interesting is that during development, this tissue actually starts as a
monolayer of multipotent cells, which are called progenitors. And in particular, these progenitors are organized in a pseudostratified epithelium, meaning that they're all attached at the apical and the basal side. What we're interested indeed in understanding with my PhD project is how neurons are generated from progenitors at the right time and in the correct proportions in order to form this beautifully layered and then functional tissue. And in order to answer this question and to understand how neurogenesis occurs during development, we use live imaging of the zebrafish retinal zebrafish embryo. And in particular, we do this because, as you can see from this movie, we can actually look at the development of this tissue. We can look at single cells and what fates these single cells will then give rise to. This is interesting because thanks to this model system, we can actually follow single progenitors in time with live imaging, and we can actually look at the cell divisions that these progenitors undergo and the fates that then will give rise to. So in order to understand how multipotent progenitors can generate different neurons, what we did is that we imaged the first neurogenic lineage that occurs in the retina. And what we found is that the multipotent progenitors transition to different types of neurons thanks to two consecutive asymmetric divisions. One first division that produces one neurogenic progenitor and another multipotent progenitor, and the second division that starts producing neurons. And what we were struck by is that these divisions are both asymmetric. And while we already investigated how the symmetry is set in the first division of the lineage, we still don't know much about the second division. So what we would like to understand with possible projects for master students, it's first of all, what are the cell biological factors that can contribute to set the symmetry of this second division? While on the other hand, there are still questions about the first division, right? So we still don't know, for example, how these progenitors enter neurogenesis in the first place, and if they do so at a certain time. So what we would be interested in understanding is if progenitors enter neurogenesis after a fixed number of cell divisions, or if there is some other mechanisms that could set the timing for neurogenic entry. So this was very general, but of course we have some few more slides, and if somebody's interested, we can go to the breakout rooms and then we can chat a bit more about this.
Thanks, Elisa. And yes, whoever is interested in these interesting lineage decisions and how we can follow them in the fish, meet us later when we're done here.
Thank you, Karen and Elisa, for your project. And now I'm passing the word to Pablo Sartori.
Is that working?
Yes, it's working.
Hi. So let me turn on the lights for a second. My name is Pablo Sartori. I'm also a group leader here at the Gulbenkian Institute. I'm a bit different from other group leaders in that I was trained as a theoretical physicist, so my research is more qualitative. I didn't prepare as many slides as the other people, but while they were talking, I took a snapshot of my website. So I also have a website. So if you're interested in my work beyond what I can fit in a couple of minutes, you're welcome to look in there. And one general question that we're trying to address in the small but growing group has to do with the self-assembly of proteins into complexes. Okay, so in the cell, as most of you probably know, there are proteins, and these proteins assemble into larger structures that are called protein complexes, okay? And one of the things that I'm interested in is how these higher order structures form and how they carry their functions, etc. And as a physicist that I am, I think that we need to be able to give some basic numerology on this topic, right? We need to be able to discuss the sizes of complexes. We need to be able to discuss their amino acid composition, their whether they're polymeric or not, the different degrees of symmetries and things like this, right? Essentially, we need to know what we're talking about. We need to have the basic variables. And once we do, we can start addressing more profound puzzles, right? So one puzzle is this puzzle that you may all know. It's called the Game of Tangram. And in the Game of Tangram, you're given these 7 pieces. And the task for you, the player, is to elaborate different shapes, such as the cat, the duck, or the rabbit, by rearranging these pieces. And one question is, to what extent things like this happen at the cellular scale, right? And it seems that they don't happen very extensively, but there's reasons to believe that this shouldn't be the case. However, it's hard to know whether it happens or not if we cannot quantify how many complexes there are and whether their structure is, what their properties are. So this numerology, not to be confused with astrology project, has two parts or 2 directions. One is a bit at the smaller scale. And the idea would be to try to understand the numbers of typical protein complexes. So for example, here you have a polymerase complex. This is a protein that a protein assembly, a protein complex that has 6 proteins in total. And among these 6 proteins, there's 5 different ones. Okay, so all of a sudden you realize there's at least 2 numbers you can give to a protein: the complex, the types of proteins, and the number of total protein content. But below you have a very different assembly. This is Atp Synthase, and it has 26 proteins total and 9 different types, right? So you can see there are differences in composition, in size. There's also differences in symmetries and so on. And the first task would be to come up with what are the good numbers to characterize protein complexes and find some automatic way of going through databases and parsing them to generate these numbers. The second part of the project has to do with what I call the large scale structure, right? So protein assemblies, like us used to a year ago, don't live in isolation. They interact with each other. And then you can ask how these interactions happen, right? So one way in which these interactions happen is because proteins are shared among different complexes. And this is also something about which we can do statistics by using publicly available databases. And this has not been done. I've done a little bit myself, but not much. So I can help master students compile these databases and I can help them analyze them. And then there's other types of things one can do, like one can discuss properties such as heterogeneity of a protein complex and then cross-reference these properties, right? So heterogeneity has to do with the protein content and the composition of complexes, but then we can cross-reference with the structures, right? And again, this is something on which I've done a little bit to discuss what are the difference between asymmetric and symmetric complexes, but I haven't really done it in much depth, and I think master students should have hopefully fun doing it. That's all I wanted to say. Thank you.
Thank you, Pablo, also for your presentation.
Yep.
And now, can you please stop sharing your screen?
Yes, I should be able to do this.
And now, Raquel, do you want to talk to us a little bit about your project and about your research?
Yes. So I'll just share the screen, and hello, good afternoon, everyone. And yeah, it's really great to see so many people interested in these sessions. So I'll tell you a little bit of an overview of what are our research interests in the lab. So my name is Raquel Oliveira, and I'm the head of the Chromosome Dynamics Lab at the IGC. Oops, sorry. And in our lab, what we are interested is really in the morphology of chromosomes, which is absolutely critical for faithful segregation of the DNA during every time the cell divides, during mitosis, so that you really acquire this beautiful symmetry that the 2 daughter cells inherit exactly the same genetic material. And of course, this can occur with some errors in this process, as for example, if cells inherit the wrong number of chromosomes, and you have numerical errors, which are also called aneuploidy. but also you can have damage to the DNA molecule itself, leading to DNA breaks. And this has potentially, it's a source of problems into the maintenance of genome integrity. And basically what we do in the lab is trying to understand the processes related with chromosome organization that really ensure this beautiful symmetric process. And we are mostly focused on three major topics. So one is what I call the gluing problem, which is the cohesion between the two DNA molecules that needs to be established from the time that the DNA copies itself during DNA replication all the way to the later stages of mitosis. And this is absolutely critical to ensure faithful genome segregation. But because if you have premature sister chromatid separation, then the cell does not have a way to know that this one is identical to this. And it will basically, it's almost like shuffling cards, and it will basically segregate the chromosomes in a random manner, thereby leading to very severe numerical errors and aneuploidy. And the second major problem we are focused in is chromosome compaction, which is how do chromosomes change their shape and become this famous X-shaped structures that are critical to ensure faithful genome segregation. Because again, it is known that if chromosomes do not properly compact or do not resolve the intertwines between the two DNA molecules, this can lead to DNA being trapped at the cleavage furrow that can, of course, lead as a source to DNA damage. And a more recent research topic in the lab has to do with the silencing problem, which is how do transcription, that it's so active during interphase, and it's actually what makes cells to read the book that is the genomic material and actually perform its functions, how this is promptly shut down during mitosis. And the reason for that is that we also have preliminary evidence that suggests that if cells do not properly switch off transcription, this can be also a source for potential errors during cell division. But now focusing more onto the, so just as a summary, and the reason why we are so interested in this is, of course, because if you have problems in cell division, this has been associated with many different types of human pathologies, such as cancer, infertility, and development disorders. And now moving into this project itself, it is mostly focused, or at least the source of the question started with the gluing part, but I should tell that we really think that the project itself can grow into any direction, not only on the gluing, but also on the packing and also on the silencing problem. And when I'm talking about gluing, I'm mostly talking about cohesin, that is actually the molecular glue that holds sister chromatids together. So it's a ring-like protein that embraces the two identical DNA molecules. And the master project that we have here proposed actually starts from prior results that we obtained in a fruitful collaboration with Rui Silva and Rui Martinho, where the major question was, can we rescue gluing problems? So if we can revert the problems that are associated with cohesion malfunction. And for this, we basically used a genetics screen in Drosophila wings, where we downregulate a cohesin stabilizer, so this SAN RNAi. And of course, the wings in this case are highly abnormal because cohesion is super essential for the cell division in the wing, and therefore defects during development give rise to this abnormal wing. And what was also interesting is that this intermediate phenotype was subject to phenotypic modulation, and we could then use it to screen in a blind manner for conditions that could either enhance the effect on the wing. Basically, we would go from a wing like this to something more severe, or what we would be interested in case of rescuing cohesion problems, they could suppress the defects that were associated. So basically, if we knock down whatever gene, this would somehow be able to rescue the defects, and flies would become more normal. So this study, and actually we have not yet covered the entire genome, so only about 20% has been covered. But after counting a lot of flies, we came up with a list. And what was reassuring is that we could find as enhancers of the phenotype, so things that make it worse, proteins that are associated with cohesin already. So this would give us confidence that the screen was actually working and we were able to have everything functioning properly. And of course, now this also isolated several proteins that are able to do what we wanted, which was to rescue the cohesion problem. And for a lot of them, we already know what they are, but there is a bunch of proteins that here states that it's not known exactly what they do. So this master project is really trying to focus on some of this that we believe are critical players for mitotic fidelity that may regulate directly the glue, but it may also impact on chromosome structure by other means. And I'm just going to give you a flavor that one of the things that we mostly use in our lab is live cell imaging. And the idea would be to perform phenotypic analysis of these identifier suppressors and uncover, how they affect mitotic fidelity, and if so, what would be the mechanism. And with that, I just want to present you the team. Of course, this is also taken before COVID times. We also don't have an updated figure. And I'm happy to discuss with you in the breakout room if you want to have some more details on the project itself.
Great, thank you, Raquel. And now, Miguel, do you want to start? So next project is Miguel Soares and Rui Martins. But I think, Miguel, you are the one that is presenting it, right?
Yeah, I think so. Let me try. Sorry. I thought I was one after. So, thank you for organizing this really interesting meeting. And it's really nice to see so many of you that are interested eventually to join us at IGC. So my name is Miguel Soares. I'm the head of the Information Laboratory. And this is us on a not so summery day. So it's a lively team with many nationalities. This is Rui, which you just heard his voice. This is Wilson, who's the student. This is Sunima, who's a student that just left the lab for Boston. And other people that I shall introduce later on. So, sorry, so we're really interested in how we protect ourselves against infection. And the dogma, I would say, and it's not wrong, is that we use our immune system, because our immune system has both soluble molecules and cellular components that are really, really good at detecting the presence of pathogens. They either detect it directly, so they have what we call pattern recognition receptors, and they detect what we call PAMPs, which is pathogen-associated molecular patterns. And these innate cells, as we call them, usually macrophages, if they detect a microbe, they activate what we call an inflammatory response. They will eventually go on and trigger what we call an adaptive response. And the whole idea is to destroy the pathogens. Right? And this works very good for viruses, for bacteria, for parasites, and that's how we survive. However, there are some diseases that this does not seem to be sufficient, or even worse, it seems to be the cause of the disease itself. So it's our immune system, instead of protecting ourselves, it's actually contributing to the disease. So classically, when I talk about this, I give the example of malaria, which is the disease caused by plasmodium infection. Millions of people infected every day, millions of people that die. Every year. The other example is sepsis, which is probably one of the major causes of death worldwide. Everything with every confounding factor. And that's triggered by a systemic bacterial infection to which we respond, but we respond so strongly that we die from it. And now, unfortunately, we all aware of COVID-19, which is a viral infection by SARS-CoV-2. in which a very small fraction of people, in percentage, actually develop a very severe disease and can die. In all three cases, the problem does not seem to be that we cannot clear the pathogens. So there is something else that we had not perceived for a long time that can explain these major causes of mortality and morbidity in humans. And that has to do with another disease, with another defense strategy, which is as important as the one triggered by the immune system, that we call disease tolerance. So disease tolerance is a defense strategy that is conserved from plants to Drosophila to humans, that aims exactly at the same as our immune system, but as one very particular feature, it does not target the pathogens. So what does it do? It actually prevents metabolic dysfunction, and it prevents the level of injury that is imposed to different organs. And by doing that, it allows us to keep functioning despite the fact that we are infected. So I'm going to show you a little cartoon for this, which was actually the first author of the article is Rui. And these are different pathogens. So this is Plasmodium that causes malaria. This is a fungi. This is virus, could be SARS-CoV-2, and this is bacteria that causes sepsis. This is the immune system with, adaptive and innate immune cells. And their role is to sense and kill these pathogens. And they do that because they sense them through immune activation. Like many other things in life, this all comes with a price. So we can sense, we can kill, but still we can cause actually stress and damage to other tissues in our body. For example, SARS-CoV-2 causes tremendous damage in the lung. So if we take that example, that might come from virulence, so from actually genes that are encoded by the pathogen, or it comes most likely from the immune response that is overt and too strong against this virus. Now, what we and others discovered is that without weaponry against this stress and damage, we can trigger what we call tissue damage control mechanisms that limit the extent of the stress and damage that is imposed. And we're very interested in exploring what we call this transcriptional stress and damage response network that I'll spend some time describing. So the output of tissue damage control is the establishment of disease tolerance. That is a defense mechanism that protects you against the pathogens, but it's very downstream. It's not really here where we usually work on it. So the stress and damage response network, which was actually designed by Rui, it's actually very interesting. This is about, I think, 16 transcription factors. Each one of them responds to a different form of stress and damage. And we're very interested we thought that there were the key factors that would explain the establishment of disease tolerance. They regulate affected genes by themselves, but they actually interact with each other to express other genes. And as you go to the interior of these circles here, you see that these core genes here are actually regulated by more than 7, sometimes 9 of these transcription factors. So we're very interested to understand what are the core genes in this transcriptional network that actually provide tissue damage control and disease tolerance to infection. Now, in a very schematic way, I brought you what several members in the laboratory are really interested. These are tissues trying to protect themselves. See, they get a little bit of injury. They release what we call alarmins. And immune cells, actually, they're not really concerned here in killing pathogens. They're concerned in sensing the damage that comes from the tissues and reacting upon it, okay? So these ones are where the stress and damage response pathways are activated. They communicate with these cells that we call macrophages, and macrophages help them to keep functioning. If this is a neuron in the brain, it's essential for brain function. If this is a cardiac myocyte in the heart, this system will be essential to maintain heart function. Now, the applications of these type of very fundamental research are obvious because We think this is essential to understand and treat sepsis that I told you is this life-threatening organ dysfunction caused by a dysregulated response to infection. It has a huge global burden, much more than cancer, actually. We see it as progressing from infection. There's an immune dysfunction that everybody agrees on. There's this weird thing called metabolic dysregulation, and that's actually what causes organ dysfunction. And we work exactly here. So this is one of the projects that we're interested in. The other system that we work on is malaria. I'm not going to describe to you. Complex disease transmitted by a vector, which we actually operate with. Goes to the skin, goes to the liver, and once it gets into red blood cells, you get all these types of disease that can develop. And we've shown over and over now that these stress and damage response pathways can operate in the kidney, they can operate in the brain, they can operate in the lungs, and they're absolutely essential to survive malaria independently of the pathogen. But what we're really interested now is to understand how immune cells are interacting with cells in these different tissues to make this disease tolerance mechanism operational. And I think I'll stop here, Anna.
Thank you, Miguel. Can you also stop sharing your screen?
Absolutely.
Perfect. Thank you, Miguel. And now the last speaker of this session is Lunes Chiki. And Lunes and Remy, they have also a project. Actually, Lunes has two projects. So Lunes, can you, I don't know if it's you or Remy who is going to present, but please go ahead.
Okay, I'm going to start and then just present the group, and then Remy will present the last part. Sorry, share. You probably see it. You see my screen?
Yes, we do. Yes, we do.
Okay. Okay, so you see it. Okay, thanks for coming to all of you, and thanks for the invitation, Anna, to present the projects. My name is Lunes Chiki. I work here at the IGC, where I'm responsible for the Population and Conservation Genetics Group. I also work in Toulouse at the CNRS in the University of Toulouse. The group, Population and Conservation Genetics Group, like any group, has a history. It has diversity in many ways. And you see in the lower part of this slide, some of the recent members. Some of them have gone, like Beatriz Muraton, who's number 3, starting from the left, who's now doing a PhD in Germany, for instance, and on. The kind of questions that we try to ask in the group are related to what's happening in this slide. So we're interested in understanding genetic or genomic data at a population or species level. And we're trying to understand it by asking questions about patterns, which is how is diversity, genetic diversity distributed in space? Is there more diversity here or there? Is there more diversity in some areas of the distribution of a species? Is it related to barriers, which may be natural, which may be anthropogenic, so due to humans? But also we try to understand processes, and to do that, we sometimes try to integrate other types of data. If we work on humans, we may be using data from linguistics, archaeology. If we work on other species or humans, we can also try to understand ecology, paleoclimatology, and try to come up with an evolutionary history. So the idea is to try to understand some aspects of that history, such as population size changes, population structure, and sometimes selection. In the group, we work rarely on selection, but that's possible. Another way of seeing it is really to see this in this slide, in which you have different types of genetic data, which are obtained at what we can call a population. That is, we're not necessarily interested in the genetics of a trait. We're more interested in the diversity across populations, individuals. And we're asking questions in two main areas. On the left, you have conservation genetics. On the right, human past demography. And often this leads us to ask questions about how to develop new methods, possibly software, to answer the questions about habitat fragmentation on the left, for instance, on conservation genetics, population decline, but also recent evolutionary events in the demographic history of human species. We use different kinds of modeling approaches in the group. So for instance, we can use spatial simulations to study habitat fragmentation, and that's the PhD project, for instance, of Gabriele Sgarlata. We have been working on social structure, that is, to what extent does the existence of social structure in some species lead to different genetic diversity levels in different species. That was the work developed by Barbara Parreira. And we also work on what we call genealogical models, which are models in which we try to understand how, when we look at genetic diversity and try to understand the genealogies of the genes we look at, how they inform us on the history of a species. When I talk about models, because we are interested in conservation questions as well, there are many kinds of models, biological models we've been working on. I mean, I have developed a very long-term collaboration with people to work on orangutans and Bornean elephants, so in Southeast Asia. In Madagascar, now it's been 15 years. We've been working in Madagascar on different lemurs, but also rodents and other species. There is a person called Tania Mignos, who was a member of the group. She's a postdoc, and now she has a position at the Universidad Nova, and she has developed projects in West African primates. We have these projects on humans, and there is finally Ines, who's been developing projects on the Sado dolphins. The projects that we proposed for this call are more related to genealogical models, that they involved all these people one way or another, even though in one of the projects it's mainly myself, Beatriz Morato, and Olivier Mazet, and I'll give the title of the project. And then there is the other project in which it's Rémi, who's been developing a very nice project, and myself. So the two projects, as a reminder, for those who saw them and maybe forgot. So one of them is comparative demography of endangered species using genomic data. So that's more related to endangered species. And the idea is to try to understand something about the demographic history of species by looking at several species at the same time. And the other project has an even longer title, which is testing the confounding impact of sampling schemes and spatial structure on signals of population size change and admixture. If you still remember the beginning of the title, that's good. So Rémi will say a few words on that, and it's related to human evolution and asking questions about how different types of model may lead to quite different kinds of conclusions. I'll just say a few words, one slide on the project that I mentioned, and if you have more questions, I'll be happy to answer in the small rooms. So in Madagascar is very famous for being a biodiversity hotspot. We know that now it's it's biodiversity highly endangered because of habitat fragmentation. There's also the risks due to climate change and human activities. All these things are not questions. Now, another type of questions is related to the past and understanding how ancient habitats may have played a role in the biodiversity we see within species or within closely related species, and how maybe ancient climate events may have played a role. And to do that, we try to use genomic data and, for instance, infer how connectivity may have changed through times with methods that we develop with people, collaborators in Toulouse. And these curves, and I won't say more about that, are results, for instance, of analysis obtained for different species, in which for each of them, we have genomic data, and we try to understand if there are periods of high gene flow, that's when the curve is high. So this is for one species. Time goes from here, recent past, ancient past. And you see these curves change, and we're trying to understand, and this is the kind of questions we would like to ask with students interested, is how do these curves that we obtain across several species, are they similar? Do they identify similar periods during which species, for instance, here you have 3 individuals of the same species, and they come from different locations. And we find that, for instance, gene flow seemed to have been high, then became very low and re-increased. And then there's a huge uncertainty on the recent past. And this kind of question is, do we find similar patterns across species? Now I'll let maybe Rémi, if you want to speak, since the slide is on.
So apparently it might close very soon, so I will go very fast. So the idea is to investigate alternative models of human evolution. You have on this slide represented on the left one classical demographic model that are developed in human genetics and human evolutionary biology, where, for instance, you have mega populations where, for instance, human individuals from East Asia are represented as one big population within which everybody can reproduce freely with any other one, which is completely unrealistic and very rough. In this kind of human evolutionary model here, for instance, in the illustration, you can see the admixture arose, which indicates the hybridation that is now a consensus in human evolutionary biology about the fact that all non-African humans are the product of a hybridation which occurred between Neanderthal and Homo sapiens sapiens. What we are doing and what the project is about is to question this consensus. Especially question it regarding what I just said previously about the roughness of the population resolution. What we try to investigate, what the project is about, is that if you integrate, if you take into consideration the fact that human populations are structured, both spatially, socially, in many ways they are structured, like individuals do not freely reproduce with other individuals living 1,000 kilometers from them. If you integrate this structure, then there are genetic signals that are attributed to admixture, especially Neanderthal admixture, which you can explain just by this structure. So we try to test models, for instance, where what is supposed to be evidence and what is given as evidence of Neanderthal admixture are not so much evidence, because we could explain those signals just by taking into account the structure in human populations. And you have on the right one such structured models, which is much more complex, because you can see that even though it's still rough, you can see that we take into every point in the African and Eurasian continents represent one population, and we integrate the complex network of gene flow which can happen between these populations and see how these spatial structure, the way we sample within these populations, how those structures can confound signatures that are attributed to other factors. So it's for now it's theoretical work, but the purpose is then to extend it further to test alternative models of human evolution.
I'll stop sharing.